Today, we welcome one of the 25 most recognized voices in precision medicine globally, a renowned international oncologist. I'm, of course, speaking about Dr. Razelle Kurzrock. Now, to give you all just a brief highlight of some of her many accomplishments. Dr. Kurzrock is co-founder, board member, and chief medical advisor of CureMatch, a company that is focused on supporting oncologists with decision support technology for precision medicine. A board member of CureMatch, a company focused on AI technology for earlier detection of cancer and heart disease. Professor of medicine, associate director, clinical researcher at the MCW Cancer Center and Genomics Sciences and Precision Medicine Center. The Linda T. and John A. Mellows Endowed Chair of Precision Oncology, the Chief Medical Officer, Executive Committee, Equal Opportunity and Diversity Officer of the Worldwide Innovation Network, or WIN for short, for Personalized Cancer Therapy, a nonprofit organization, and also an adjunct professor at the University of Nebraska. She has published over 850 peer-reviewed manuscripts in such prestigious journals as the New England Journal of Medicine, the Journal of Clinical Oncology, Lancet Oncology, Cancer Discovery, Cancer Medicine, and so many, many more. And today she shares with us advances in matching patients after next generation sequencing for personalized cancer therapy. And without further ado, it is my honor to welcome Dr. Kurzrock. Thank you.
Well, thank you for that very nice introduction. Are you able to hear me?
Perfect.
So I appreciate that very nice introduction. And I'm going to wear my entrepreneur hat today and talk about CureMatch, a company that I co-founded with technology. And that was initially taken out of, bought out of UCSD. And it was really based on my experiences as a department chair at MD Anderson, leading an early phase, very large early phase department, seeing what matching patients to drugs in a proper way could do for cancer. And later at the University of California, San Diego, leading the precision medicine program. And again, as next generation sequencing and other omic technologies became more sophisticated, seeing again the transformation that was beginning to occur in oncology, but also the complexity that was being introduced that really needed a solution in order for physicians to be able to properly address it in their individual patients. So, cancer complexity is really the issue that CureMatch addresses. This is a challenge for doctors, patients, and healthcare systems. It's meant to fill the unmet needs gap and to address precision treatment with the best matched, optimized matched drugs. And this should have an impact, not just on clinical outcomes, but also our ability to optimize treatments on cancer clinical trials, and also optimize our financial resource use. So I'm going to go through what we do. And this is a bit of a hybrid talk where I'm going to talk about how CureMatch is translating this into the clinic, but also my academic background and some of the issues that I feel are really important from an academic standpoint. So what if every patient with metastatic disease is different? What if every metastatic tumor is different? I've lived in California for a while now, and I was at MD Anderson Cancer Center, which is in Houston, both very warm, but I grew up in, and so I know a little bit about snowflakes, and snowflakes have individual crystal structures that are unique. Every snowflake is different, and every snowflake is very complicated. And we now think about metastatic cancers as malignant snowflakes. Each one is very complicated, and each one is different at a genomic and certainly at a multi-omic level. And that means that the models that we've used to treat cancer in the past are not the best fit. And what we really need is an N of one approach, looking at every patient as an individual and understanding the complexity of their tumors. And I'm just going to give you this example here. This is next generation sequencing on metastatic breast cancer. I've overseen next generation sequencing probably on more than 21,000 patients. Of course, they don't fit on this slide. So we're just going to use a few patients as an example to show you the points. So this patient and this patient, number 42 and number 25, both have ERBB2 amplification. And we can target ERBB2 amplification with a variety of different ERBB2 or HER2 targeting agents, such as Herceptin, and so forth. But if you look at the rest of the molecular portfolio of each of these patients, we notice two things. One, it's complicated. There isn't just one alteration. Each patient has multiple alterations. And second, the alterations in these patients, in these two patients, are dissimilar. They're distinct in each tumor. And that means that what we usually do in giving these patients the same regimen, just because they have something in common, and that's the ERBB2 amplification, doesn't seem right from a scientific point of view. Now, they both need an ERBB2 targeted agent, but if we're going to do this properly, we need to target the rest of the alterations in these patients individually, and give each of these patients a different regimen that might have something in common, but it shouldn't be that the entire regimen is the same. So the idea is to get the right drugs to the right patient at the right time. I mentioned that at the genomic level, every metastatic tumor is unique and complicated malignant snowflakes. And as we develop new technologies, transcriptomics, immunomics, and so forth, I think that is becoming even more true, even more complexity, and definitely each tumor is unique. Therefore, metastatic disease requires customized, individualized combination therapy. For the most part, single agents are not going to be optimum. And I want to mention that the pillars of precision medicine are genomics and immunotherapy, and they may be viewed separately, but they're actually married to each other. And then I want to go further and say that all types of therapy actually can be individualized. So what we look at, both in my academic life and from the point of view of CureMatch, is really all types of therapy and what is the best match for patients. So let's go over where we are with cancer patients. Most cancer patients are diagnosed in later stages, which impacts survival rates. And even though we're making a lot of progress, many metastatic cancers have 2-year survivals that are still less than 50%. In some cases, a lot less than 50%. And the impact economically, we all know, is across the world in trillions of dollars. And one of the challenges is that if we look at all the possible, for instance, 3-drug combinations, there's about 4.5 million possibilities. And so it really is not possible for any oncologist, or really it's beyond human cognition, to put all of this data together, especially when we layer on top of it all the hundreds of thousands of possible genomic variants. And then if we layer on that RNA and protein and so forth. What we do, even in precision medicine, is akin to what my good colleague, Dr. Sabaya, calls the whack-a-mole game. We are trying to impact one genomic alteration at a time with targeted therapy. And as we do that, resistance quickly develops because the patient has or develops other genomic alterations. And so we think that this was a good starting point, but it's not ultimately where we want to be. In Boston, San Francisco, and San Diego, these are major entrepreneurial centers, and for biotech is especially important in San Diego. And so we helped found this company to really give the power into identifying novel optimized combination therapies with really robust algorithms that could input data from multiple sources, all the data that's known about drugs, the data that is known about omic variants, and look at optimized combination therapy for each patient. So let's go over the way I've practiced oncology and the way I think everybody has practiced oncology for the last 100 years. Oncology is drug-centered. It's not the patient that is at the center, it's the drug regimen that is at the center. And that's true in both clinical trials and clinical practice. And patients may have different alterations. Patient 1 has A, B, and C, patient 2 has A, E, and F, and patient 3, A, B, and F, but they get the same drug regimen. So how we do this is we try to find a common feature between patients. For instance, the type of cancer. Do they have lung cancer? Do they have breast cancer? And then we give them all the same therapy. Initially, these drug-centered trials really concentrated on histology-based or site-based therapy. So we looked at patients and we said, are you a lung cancer patient? Okay, these are the regimens that we're going to give you. Or a breast cancer patient, these are the regimens we're going to give you. And of course, that is still the basis for a lot of what we do. But there's been another direction that has happened, and that's looking at a different type of commonality, and that's a genomic commonality. This was almost unimaginable 10 years ago, but now we have 6 tumor agnostic approvals, for instance, for NTRK alterations. So if you have an NTRK fusion, it doesn't matter what solid tumor you have. It doesn't matter if you have a colon cancer or breast cancer or a renal cancer. You can get a drug, larotrectinib or entrectinib, that targets that fusion. So this has been a major breakthrough, but the paradigm is still in a way, the traditional paradigm. And the paradigm is that every patient gets the same drugs regardless of their molecular differences. In other words, it's still drug-centered. So as long as patient 1 and patient 2 both have an NTRK fusion, it doesn't matter what the rest of their alterations are, how many alterations they have, and how different the alterations in each patient's tumor are. They're both going to get an NTRK inhibitor. Still, targeting the genome rather than only looking at the site of origin, I consider a major move forward. But I don't think it's the final move on the road to treating cancer. So the way we have treated cancer traditionally, the drugs are at the center of the universe, and then we find patients both in a clinical trial and clinical practice setting that fit onto the drugs. Now, what I believe that we need to move to, and this is the emphasis of CureMatch, and also has been the emphasis of the clinical trials that I've conducted over the last 5 to 7 years, is a patient-centric approach. And that means that it's not the drugs that are at the center of the universe, it's the patients that are at the center of the universe. And each patient has drugs that are customized to them. So we don't find the patient for the drugs, we find the drugs for the patient. And we do intense interrogation with molecular and immune marker matching for each patient, and then customize the therapy for each patient. So both of these patients have marker A, but patient 1 has B and C as well, and patient 2 has A, E, and F. And so they get a different combination of drugs that is matched to their specific alterations. And that moves, instead of the drugs being the center of the universe, the patient is the center of the universe, and we match the drugs to that patient. Now, I want to say that this is really new in oncology, but it's actually not new in medicine. It turns out that if you really think about it, oncology is the exception to the rule, because in medicine, we all practice patient-centric care every day. So if we have, the average patient walks into the oncology clinic with 8 different drugs that have nothing to do with cancer, because a lot of these patients have many comorbidities. And if 2 patients walk in and they both have rheumatoid arthritis, that doesn't mean that they get the same drugs, regardless of their other conditions. So if patient 1 has rheumatoid arthritis, congestive heart failure, and depression, we give that patient an anti-inflammatory drug, a beta blocker, and an antidepressant. And if patient 2, who also has rheumatoid arthritis, but has an infection and hypertension, that patient still gets an anti-inflammatory drug, but the rest of their drug regimen is different than patient 1. They get an antihypertensive and an antibiotic. So matching patients with customized combination therapy, while it is new for oncology, it's not new for medicine. In fact, I want to reiterate that oncology is the exception to the rule. We practice individualized combination therapy as physicians every day outside of oncology. So the next stage, we believe, is redesigning cancer strategy, and that's N of 1 trials. And that means instead of using a consistent drug between patient or drugs between patients, even in the presence of different molecular profiles, which is the traditional way, we should be using a consistent strategy, a robust algorithm, but allow different drugs. And that's really what we've done with CureMatch. So one of the issues with doing this, and perhaps the most important issue, is the heterogeneity of cancer, how complicated cancer is, how many drugs there are, how many possible combinations. I mentioned 4.5 million possible 3-drug combinations, if there's 300 drugs that could be used in oncology. And then layer on top of that the number of variants, and that's just genomics, never mind transcriptomics and proteomics and immunomics. And this is really beyond human cognition. And so we founded CureMatch in order to develop the most robust algorithms so that the best matched combinations could be figured out for each tumor. Where is this important? I think that this is, personally, I think this is important for any patient with cancer. But I think it's especially important where an oncologist wishes they had the expert advice at a molecular tumor board. Although I will say even molecular tumor boards need this type of decision support. My own experience leading molecular tumor boards without this type of decision support is that people are sometimes just sitting there Googling, what does this alteration do and how can we combine these? And that's probably not the optimal way to access the tremendous amount of data that is really out there in order to optimally address each tumor. So what we try to do is have a customized oncology report for precision medicine. And this puts together all the information that are necessary for the patient. So we use any type of NGS sequencing, but not just genomics. It can be transcriptomics or immunomics. And we look and score standard of care therapies, immunotherapy, chemotherapy, hormonal therapy, gene-targeted therapy, therapies that may not be usually used for cancer, but actually target alterations found in cancer patients. And the reports are optimized for NCCN and FDA approved drugs, but of course include as well drugs that are off label or clinical trials and so forth. So how does the platform assess thousands of options? These are platforms that are set up to be able to digest almost unlimited amounts of information. And that information includes all the things that a drug may do. So remember that a drug, a targeted drug, may be marketed for a specific alteration. But that especially small molecule inhibitors, that's a little different from antibodies, it may actually target 25 or 30 different targets. So that information is difficult to access for most physicians and caretakers, but all of that information is included in the platform. The IC50s, which can be variable anywhere from 1 nanomolar to micromolar range, all of that is taken into consideration. What drugs go together well, what drugs do not go together well, and so forth. And out of that, we have developed a scoring system with scores of 0 to 100%. The scores do not indicate the chance of response. The scores reflect how well the drugs are matched to the patient alterations. So we feel that this is the next step in being able to put together data that is just increasing in complexity as we get more and more information that is available, and information that was really not imaginable 20 years ago, really not even imaginable a decade ago, and moving even quicker with at really a breakthrough rate to bring in more information about individual tumors, so we understand the microenvironment, so we really understand all aspects of the tumors and being able to put that together. One of the questions that comes up is, well, what about financial value in drug therapy once you start putting combinations together? And where does this algorithm, where does this type of technology apply? So I think today it really applies most in metastatic tumors, especially if patients are out of standard treatment options, refractory tumors. But my own personal feeling is just like NGS, which I believe really applies to all patients with cancer. I know that that's still debatable, but that's my personal feeling. I think wherever you do complicated interrogation of a patient, technology like CureMatch that really can put all the data together is really essential. And then the financial issue of giving combination drugs, which a lot of people ask about. Drugs in oncology are expensive. If you give one drug, that's expensive. Never mind if you give 3 drugs. As most oncologists know, we often give combination therapy already. It is very common to give more than one drug, sometimes 3 or 4 drugs to cancer patients. But the difference here is that these drugs are very matched to the cancer patient's molecular profile. So when we look at the dollar value per CureMatch point, with the CureMatch points reflecting the degree of matching, we see that the dollar value is best with higher degrees of matching. So it's not a question of yes combination or no combination. It's a question of best combination and best value, best financial value there as well. Now, I want to say that I've talked about individual patients because that's really where my heart is. But this can actually be applied and translate very well for clinical trials as well. So just as we can choose based on these complex augmented intelligent based algorithms, the optimized match for each patient, we can also choose that for patients in clinical trials or populations for clinical trials. Which population is most likely to respond to a specific experimental drug, knowing all the properties of the drug and knowing the sequencing of that tumor. Just like for individual patients, we can figure out what's the best match. That really applies to experimental drugs and clinical trials as well. So I think this can really enhance our ability to move drugs forward in the clinical trial space. So the next question is, how do we know this type of thing works? And I think that's a really good question, both from the CureMatch side and from my academic hat side. We've actually done a lot of studies, and these are studies in the clinic. Some of them are real world or retrospective studies, and some of them are prospective clinical trials. And I'm just going to give you a few examples. So this was a retrospective analysis of exceptional responders. Those are patients that are published in the literature that have had great responses And really, a lot of times we really don't understand why. So we went through and curated all the data for the time period in the literature. And what we found was, and we picked patients that had exceptional responses and where we had a molecular profile available. What we were able to show is if we calculated the CureMatch score, the exceptional responses had the highest CureMatch score. And when we looked at these patients' prior regimens, which they had failed, those regimens had a much lower score. Furthermore, we instituted what I think was a really novel paradigm for our molecular tumor board at the University of California, and as well for a series of prospective therapy trials called iPredict. And this is the molecular tumor board paper that we published in Nature Communications, 715 patients with metastatic cancer. And we were able to show that patients that had a better matching score did significantly better, about a 50% response rate versus about a 30% response rate for patients with a lower score or an unmatched score. And the same for progression-free survival and overall survival. And then with Dr. Siklik and Dr. Cato at University of California, we also did the prospective iPredict study, which we published in Nature Medicine. And I think this was really the first trial that prospectively gave customized combination therapy. And what we can see is that matching patients to drugs isn't a yes or no answer. It's really the degree of matching that counts. And so patients that had a degree of matching of over 50%, which was an over 50% score, had a response rate of 45%. Patients with a less than 50% score had a response rate of 16%. And I think the survival differences are the most remarkable. These are highly refractory metastatic cancer patients. At 2 years, the overall survival for the well-matched patients is over 60%. And for the poorly matched patients, the 2-year survival is less than 20%. And the other thing that we found is that there's no inflection point. The outcomes are linearly related to the degree of matching. And what we used for these prospective trials, which was a matching score from which CureMatch, as I mentioned, came out of UCSD, which really was the foundation for the CureMatch algorithm. We've also looked at real world data that is becoming available and publicly available, where we know molecular profiles and we know the outcomes of patients. And when we look at survival probabilities, we just submitted this ASCO abstract. The p-values are very significant for the algorithm being able to predict which patients will do better. The abstract was a pilot study. We now have data on well over 300 patients with the results remaining even more robust than what I've shown here. Higher the score, the better the outcome. The idea here is to synthesize complex data and make it easily available for optimizing combination therapy or monotherapy for patients with cancer. Now, I just want to finish off with a question that I often get, and that's about how do we know the right doses for these patients? So I'm a phase 1 trialist. I probably ran the largest phase 1 department in the world at MD Anderson. And I want to address what I think is what we did at UCSD and how we need to be thinking about dosing patients. So phase 1 trials, which decide on the dose for patients. They're usually 3+3 design, which is 3 patients per dose level. And then there's some accelerated designs, which are 1 patient per dose level. The maximum tolerated dose, which determines the approved dose or the dose that generally we go ahead with, is determined on 6 patients. And these trials take at least 1 to 2 years to complete. And I'd like to say that 6 patient is probably very few in order to determine a dosing regimen, and taking 1 to 2 years to complete, that's really a long time, especially when we consider that if there are 300 drugs in oncology, as I mentioned before, there are about 4.5 million 3-drug combinations. So if we're going to do 4.5 million phase 1 trials, that's going to take over 1,000 years to figure this out. And so we really need to be thinking about this differently. And this is the solution that we came up with in the iPredict trial that was published in Nature Medicine. And we were able to very safely give the drugs, was monitored by an independent data safety monitoring board. And in fact, the toxicity was less than for patients who got standard of care. So the solution, in my mind, is instead of interpatient dose escalation, which we do in a classic phase 1 trials, when we try to put drugs together in oncology, we should perform intrapatient dose escalation. We start patients at a lower dose and we escalate them to the dose that is most tolerable for them. And there's a lot of different rules that you can have for putting drugs together. Just like outside of oncology, as physicians, we put drugs together all the time. And what we do is we start novel combinations at lower doses in individual patients. We see them weekly and dose escalate to tolerance. There's a bonus benefit here, individualized dosing. So it's not just individual drugs, but individualized dosing. Examples, 80-year-old and 20-year-old are unlikely to tolerate the same dose. And yet, what we come up with out of a phase 1 trial is a dose on 6 patients, which is then given to everybody. And yes, there is milligram per kilogram dosing, but a lot of drugs are dosed at a uniform dose, 600 milligrams by mouth twice a day, or 300 milligrams by mouth twice a day. If you think about that, how can that be right for both the 80-year-old and the 20-year-old? How can that be both correct for the 300-pound man and the 100-pound woman? And this just, should these 2 patients receive the same doses? The person on the left, I found him on the internet. He's an Olympic level athlete. Yes, he has cancer. And then this elderly, frail gentleman, in the way we do trials, both of them would get the same dose. And that doesn't really make sense. But if we do intra-patient dose escalation, that means that we start at a lower dose and see the patient weekly and then escalate to tolerance. We give not only the right drugs to the right patient, but we give the right dose to the patient. And what dose does she get? One of my favorite in your face ladies, but again, individualized dosing. So key takeaways. The CureMatch system, we founded it really to strengthen the algorithms, to make the best possible algorithms world-class. I personally think that they may be the best in the world, that unravels the complexity that is beyond human cognition, that has been introduced by next generation sequencing of tumors And remembering that next generation's genomic sequencing is just the tip of the iceberg. We're layering on top of that RNA and protein and immune status and so forth. And this can help with decision-making for individual patients or decision-making for clinical trials to find the best patient population that may benefit optimally from a new drug in clinical trials. So I will stop there, and special thanks to the MedNews team, especially to our patients and the precision medicine clinical teams that I've worked with over the last decade, and happy to answer any questions.